Welcome. So we'll begin with this incentive. So about 10 minutes. Let me open that up now. Okay, go ahead, please. That makes it 12 to come. We just started. 12:06 to 12:16. This is fast, of course. No, this is not fast. This is fine. I think that is 3 minutes. 12:16 will finish according to this. 12:13 according to that. Okay, that's it. Good. So we just began. You have 9+ minutes. Good. Okay, I've opened up the next week's thing. Next week's thing would be next topic. That would be the last topic. After which your presentations would come in. Presentations are from 15th onwards, right? But that is 2 weeks exact. Today is Monday. Yeah, 15 days from now. Yeah, or rather, on 15th, you will start your presentations according to the schedule we have drawn up. We'll do things. Yeah, some of you wanted more games. Thanks for your input, by the way. Thanks for him. You set up the questions, is it? Yeah. Oh, very nice. Good. So you gave them a structure and then you asked them to be free. Very nice. Thank you. Mhm. Oh, okay. I didn't know. She didn't tell me that. Good. Thanks. That is good that all of you have given feedback. Most of it seems fine. One wanted some more games, and we'll have at least one more definitely coming up. I think the next class is a game class, so please turn up, have fun. No marks anyway, and then we'll see. Yeah, Friday. Let's see how it goes today. If it goes fine today, let us see. Friday can be off for these two. These two are, by the way, if you have not done systems biology, they are intense papers. I don't expect you to understand the whole thing and so on and so forth. As long as you can get an import of what is there, it is fine. Of the second paper, the first paper is more of it's a 24 years old paper or 25 years old paper. Paulson, that's just given to you as a means of familiarizing yourself with what systems biology is. Okay, it was written that long ago, so not everything is covered there, but reasonably comprehensive. He has written a textbook also in 2006, I think, which is a good textbook, which people use as a textbook for their courses and so on and so forth. So Paulson has done quite a lot of work in the systems biology field, and so his nature biotech paper, I think nature biotech paper I've given us some sort of a primer in the area of systems biology. Three of you have done the course, so for you it is revision. For you, you can spend more time on the second paper, which is our paper. The others will need the time to pick things up. You may need to read other things and so on and so forth to make sense, especially the first paper. And then second paper, I don't expect you to get. As long as you get an idea of what's going on, it is fine. That's the whole point about this. It's rather involved. Okay. Yeah. Any questions, comments before we go forward? So this, as you know, is a part of flipped learning. I need to be aware of it being recorded also, so I need to keep repeating this from time to time. where you are given the material to read, and then we start discussing at a different level, so your appreciation is much better and so on. Just to make sure that you have read it, it's a part of the flipped learning strategy that we have a small incentive. That's the whole point. And we have made it open notes. It's not usually open notes, open book, but I've made it open notes, open book because I see it as a means of or as an opportunity to learn better. So that way it is fine. If I can also make it discussion based, but then it becomes a little too off and the incentive goes away. So incentive to read goes away, most importantly. So that I'm trying to avoid. Any questions or comments? Okay, this is a work on cancer. We began the work sometime in 2011. Professor Karnagaran has been my collaborator in this area. This again was around the time when I was getting out of administration. At 2008, I finished up our department thing, 2005 to 2008. And then I was at IIT Hyderabad, SAFE, and so on and so forth. The SAFE thing I finished up in 2013 Jan. And that's when I decided I will, if something comes to me, I'll do, otherwise I'm not pursuing administration wholeheartedly. Let's see how it goes. I was kind of very open about it. And then I said, if it comes to me, I'll certainly do it. I can do it well, but may not be my passion. So that's when I started looking at all these things, cancer research, even the seeds for the next topic were sown at that time. What else, what other big thing can be done, and so on and so forth. In research was sown at that time. Teaching was always there. I was always doing something or the other. I didn't have to do much to get into that. Whereas research, I had to put in a lot of things because money is needed, people to do the work are needed, lab is needed, and so on and so forth. So I needed support for cancer cells, essentially maintaining them, the entire cell biology part of it, molecular biology part of it, which changes all the time. So I've made efforts to pick it up when I was at IIT Bombay. Then I realized it is changing fast. I just should collaborate, not worry about being at the forefront of everything that is known. It's just not possible. And that too, it's not my main area per se. So that way, Professor Karnagaran has been the support for all this cancer related work. And this is some sort of a summary of the work done. As you already must have picked up, reactive species play diametrically opposite roles in cancer. At high levels, beyond homeostatic levels, they can turn normal cells into cancerous cells. Whereas at much higher levels, they actually kill cancer cells, and that's the basis for almost or most of chemotherapy and all of radiotherapy, and that's the essential basis. Maybe immunotherapy not directly, but maybe indirectly. So this is the figure given in one of the papers there, Galadari review paper. This is the normal cell and this is the cancer cell. The green one corresponds to the basal ROS or homeostatic ROS, if you want to call it so. This is yellow is drug induced ROS, and there's a death threshold beyond which it kills the cells. The ROS kills the cells, and the basal ROS happens to be higher in cancer cells. And therefore, if you induce ROS through chemotherapeutic drugs or other means, then it goes into the death threshold, and that's how it kills. Whereas the normal cells are able to handle it. And this is the general understanding here. People accept this view, and I've seen this in many places, so it seems to be fine. And as you also would have picked up, a lot of things induce reactive oxygen species in cells, and we significantly contributed to the understanding of this, that many different stress factors actually operate through reactive oxygen species, and therefore reactive oxygen species we proposed as an indicator of the stress levels in cells, the very clear indicator. So here this paper Gupta et al., this is not given to you, I can give you the reference, it's just a review paper. Pollutant, virus, tobacco, smoke, chemo, diet, radiation, I cannot read from there, xenobiotics, all of them are extracellular sources, and many more are extracellular sources of intracellular reactive oxygen species. Whereas you all know that ROS results from processes in the mitochondria, ER, peroxisome, phagosomes, and of course the NADH oxidase, which by this time was known to be a good generator of, no, actually that was known. The part about shear and NOx was not known, but NOx being a source of superoxide was well known. So all these are intercellular sources, all these cause ROS. And the reactive species, mainly reactive oxygen species, promote pro-tumor events such as tumorigenesis, in which the RS activate the oncogenes or the tumor causing genes. They inactivate the tumor suppressor genes, and they activate various intracellular signaling cascades that lead to cancer formation. Angiogenesis, formation of blood vessels. The reactive species regulate endothelial cell formation, migration, and tube formation. That way, they affect angiogenesis, or they determine angiogenesis. Metastasis, you all know, it's movement of cancer from its primary source to other sources. Reactive species are involved in regulating molecular events important for intravasation, metastasis, and extravasation. Intravasation is getting into the bloodstream, metastasis moving in general, and then extravasation getting out of the bloodstream. All these events are regulated by reactive species, and even chemoresistance is supposed to be somewhat regulated by reactive species, although the paper was not too clear on that. RS are involved in causing ineffectiveness of existing drugs. Whereas at high levels, they are involved in the following anti-tumor events also, which involve apoptosis, autophagy, necroptosis, ferroptosis, and so on. Okay. You all know what these are. We will not get into the details of these. These are all cell death events. Okay. Any questions so far? So as long as you pick that up, major part of the thing is done. What the world says about reactive species and cancer, in some sort of a nutshell, in the context of this course, that's fine. Ferroptosis, ion mediated cell death. See, essentially Fenton reaction. Fenton reaction, that this is quite a recent, what should I say, recently popular term, recently in the sense for the past maybe 10 years or so. And this is the death caused again by reactive oxygen species generated by the Fenton reaction of iron and hydrogen peroxide, hydroxyl radicals, and so on. Somehow it has become a class of its own to study. It's essentially apoptosis, or it could, if at high levels, it is necrosis, but caused by iron, and maybe there are other reasons why they want to study this separately. I haven't gone into this. If you ask Subhashri, you want to add something to that? If you ask Subhashri, she's a fan of ferroptosis. She done so much of work on ferroptosis. In fact, the next paper that you read will have her entire entire passion on ferroptosis on display there. That was entirely her own. We want when we first wrote up the paper. Well, I'll tell you that story later. You may want to look at that. I'll tell you this. Come back to the story later in that context. It makes much better sense. Mhm. The what happens is when the same drug is used again and again, its effectiveness goes down because of the reactive species activating various aspects of the cell that cause that reduce the effectiveness of this drug in general. I don't know the mechanism, and this is also a new aspect just mentioned in the passing in that review paper, and therefore I thought I'd add it also, and maybe and possibly also chemoresistance. Chemoresistance is essentially how it does it, and so on and so forth. I haven't read. I'm sure it is known, but I haven't read. And there is a good acceptance that reactive oxygen species are involved in mediating chemoresistance. How? I don't know. Yeah. When it gets converted or scale, scavenged. Okay. Hydrogen peroxide is converted to water by catalase. Oh, in that particular cascade there. Um, this thing, they are peroxides. Peroxides in yeah, they also act on hydrogen peroxide. Yes, peroxide peroxidases. Yeah, they are peroxidases. Glutathione peroxidase is the one that inactivates hydroxyl radicals. It is specific for hydroxyl radicals. I mean, rather, it can do the hydroxyl radicals also. It's not catalase is only for hydrogen peroxide, whereas hydroxyl radical conversion into less toxic forms is done by glutathione peroxidase, GPX. And if you go back into the antioxidants setup and look at it, that's one aspect. The other, okay, that was a question. The other thing that I perceived, which I thought I'll clarify, maybe it is not your question, but I'll clarify it anyway, is that all these reactions are happening simultaneously, right? It's not a sequential set of reactions and that's the only way it happens. No. Okay. So, since all these reactions are happening simultaneously in a network form, you cannot avoid one and think of a serial occurrence of the various different species. That is, so you'll have to look up, you'll have to think of it in terms of rates rather than in terms of one species getting converted to the other. So that I thought that was a question, but no, your question is simultaneously. Yes. Oh, that is also part of the question. Okay. Yeah, that's what I suspected. Yeah. Separately. The diagram is more of an indicative thing. It doesn't, it was not meant to explain the various reactions. Maybe if somebody had spent more time, they'd have made it less confusing according from this point of view. But they all have a role to play in converting hydrogen peroxide into other compounds. They all happen simultaneously. In other words, some part of the species gets converted by this enzyme, some part of the species gets converted by the other enzyme, and so on. So, and then interconversions happen all the time. So that's the reason why it's good not to follow one molecule. Then there's a confusion. So follow a species, talk of rates, you'll be fine. Is one of the enzymes that one of the important enzymes. Yes, very important enzymes. There are a lot of correlations of catalase, hydrogen peroxide, and so on and so forth. I'll have to look at them a little more carefully because again, it's not a very specific antioxidant for hydrogen peroxide. It can get converted into by so many different enzymes, and these also participate in other reactions and so on. So not too specific there. Yeah.
So when you talk about the threshold loss above which it kills, I mean, it goes under. So while administering that or weakness to make it reach the threshold, during that process, other detrimental effects can happen, like metastasis. Because we know ROS induces, I mean, it facilitates metastasis and all the other things that we saw.
Yeah. In principle, yes. But what what other things are needed. See, it is known that these facilitate, but these may not be the only causes, right? In other words, if this is high, that is also high is known. But what else need to be satisfied for metastasis to occur? Metastasis is a later event. Before it reaches that part, maybe, I don't know, I'm trying to guess here, but I don't know enough molecular biology to talk about it and talk about it intelligently. In general, whatever you're saying is true. Everything is going to happen pretty much simultaneously. However, in terms of metastasis, it seems to be an outlier kind of a thing. That is the cause of 90% of cancer related deaths. Primary tumor, the occurrence or the incidence is quite low because you can take care of primary tumors. It's only the metastasis that contributes to 90% of cancer related cell deaths in all cancers. There are variations, of course, some are much more virulent and so on and so forth than others. In other words, you need a lot more in-depth information and insight to be able to make a call. And in general, the philosophy is choosing between the two devils or many devils, right? So that all modern medicine is that, in my opinion. So, any other question? Okay, so this is some background. I think one paper was given to you. This is our work. Steffi worked on gut microbe. The story is something like this. The story is very interesting. Actually, Prerna wanted to work on gut microbes, and we said, let us first understand things on E. coli or something like that, which is a standard organism, and then we'll get to a more specialized organism because we don't know much about the gut related aspects. Whether it will be easy to grow and so on and so forth, all those things we pick up with E. coli. She got an E. coli strain from one of the sources. Since it is being recorded, let me not tell the source. I can tell you that later. Right? She got it from that, worked on it for 1.5 years. We had a lot of data. Then for some reason, we did the 16S RNA analysis. Then she came running to me one day and said, "Sir, there's a major problem." I said, "What happened?" The 16S RNA we did, this is not E. coli at all. How can that happen? I sure this thing, all the samples were fine, reproduced. It's not E. coli. What is it then? Then it turns out to be Enterococcus durans. What is that? That's one of the, luckily, it is one of the representative gut microbe organisms. She wanted to get to that kind of thing much later by a complete unanticipated turn of events. And for some time she was wondering, what am I going to do with my PhD? 1.5 years of work, a lot of work to characterization, this, that, and all that, experimental work. She figures this out. And then I said, fine, we will change the topic. We'll move it to E. coli. We'll figure out some way of doing this. 1.5 years is too much. That took a lot of work had gotten in by then. Just changing it for this. I was all set to change the thing. Then after digging, she came back and said, so this happens to also be a representative gut for the gut microbe. Great. Then the story completely changes now. We get back to our original story, you go ahead with that. And then Steffi got interested in that because she was looking at NADH redox ratios, which is ratio of NADH to NAD plus NADP and so on and so forth. Essentially the hydrogenated form with the total and things like that. And NADH redox ratio or NADH ratio is a representative of the redox status of the cells and things like that. So she was trying to figure it out for her system, which is an algal system. Then she said, I have this also, why don't I do this? And that's how they got together. They worked this out. And this is, since Steffi was doing the work predominantly, she took the lead, she did all this, Prerna provided the support. And that's how both of them are authors on that paper. So the thinking was something like this. This is completely Prerna's thinking. I think I mentioned that she came up with her idea for the PhD. Thinking of something like this. Gut microbiome was becoming hot at that time, 2015, I think. So she said that gut microbiome also plays many important roles in cancer, such as they synthesize folate, which can prevent cancer, especially in colorectal cancer. And the cancer cell lines that we are working on were colorectal cancer cell lines, CRC something. Which cell line was it? Nobody over here would know. HCT116, yeah, that's a CRC cell line. They regulate the host. In addition, they regulate the host antioxidant responses. They determine the effectiveness of traditional anti-cancer therapies, and are suspected to play a role in cancer immunotherapy also by modulating the host response. And Prerna's thing was this: during chemotherapy, you, especially when you take it through the oral route, you cause a lot of oxidative stress to the microbiome, and if it's playing a role in cancer, then we need to look at things in a balanced fashion. So it's good. Starting point is good, and to understand gut, her initial experiments are all in E. coli, and then she's going to figure out some appropriate thing and switch. By that time, she had already had the data for enterococcus, so she could directly use them, and that's how this thing came about. A part of the work is given here, her work, and this is Steffi's part. The redox ratio NADPH to NADH is an indicator of cellular oxidative stress. So they found that essentially redox ratio decreased with oxidative stress, and also the folate. This is folate. I think the open bar is folate. The redox ratio is the dark bar. This is the control with menadione, which induces reactive superoxide. This is hydrogen peroxide, and this hydrogen peroxide plus an antioxidant. In that case, I think mannitol was used. Yeah, mannitol was used in the literature, and so she used mannitol. So it is sensitive to the oxidative stress in the cells, the folate levels, and also oxidative stress reduces growth as expected. And therefore, after a lot of studies, the conclusion was that we need to carefully consider the induced reactive species and the resultant oxidative stress in the gut microbiome by anti-cancer drugs when you go ahead with those therapies. Then I think we had already looked at rhythms. Excuse me, I cannot take this now. We said we looked at rhythms in the context of microalgal cells, right? That's when we started looking at rhythms. This was Ranjani's work, and again, rhythms was entirely her part. She brought the rhythms to me. She in fact, she started talking about it with data. That's how I came to know that she was working on rhythms. She had figured out how to analyze and so on and so forth by talking with others and things like that. So it made sense at that point in time. Therefore, we went ahead with rhythms. The understanding was a little different. And then when Uma started her work and we needed to focus her work, she had taken breaks and all that. So we had to focus her work. So we said we'd look at rhythms in the context of cancer cells. That was microalgal cells, cancer cells, and let's see where it goes. Turned out fine. The idea is something like this. If you look at superoxide concentration, SOX in this case is superoxide, versus time in even in control cells, there is, in other words, there is an endogenous rhythm. Even in cells to which you haven't done anything, there is a variation in the level with time. Okay, there is some sort of a rhythm here, time-based rhythm here. So there is an endogenous rhythm. From past analysis, sorry, let me cut this out. From past analysis, past is a methodology and a software to analyze this data based on time series and so on and so forth. The frequency turned out to be 0.042 R inverse for this case, and therefore the time period was about 24 hours. So it is a circadian rhythm, circa approximately dn a day, so circadian rhythm. And the basic idea was this: in response to a suitable stress, the reactive species values vary over a range. Therefore, if you are measuring a 1-point value, there would be variations depending on the time that you measure the value. Okay. However, what we found is that there is a pattern with time of those values, of the values it takes, or how it responds to a certain stressful situation, with a variation seems to have a pattern. And that pattern is certainly the rhythm. And if there is a rhythm, if there is a variation, if there is a pattern in response to something, it's better to understand the pattern characteristics rather than the individual values. Okay, that's essentially something like the systems biology justification, right? You need to understand the interrelationships between the various different reactions rather than understanding them separately and so on and so forth. Therefore, it is more meaningful to understand the rhythm variations with stress rather than the variation of values at different time points. So, that is the reason for looking at these rhythms. So, we look at the rhythm characteristics such as frequency and amplitude and so on and so forth to make better sense or make more comprehensive sense of the effects of reactive species in this case. And that's how rhythms are. Okay. What we found was that the superoxide rhythm gets reset with Minadion. You saw the graphs in the paper. This is untreated control. This is 3 micromolar Minadion. In fact, the paper has many other concentrations also. And with all the concentrations, we could get a certain relationship between the time period of oscillation and the Minadion concentration over the 5 or 6 different values that we had by regression. Essentially, by regression, we got this. Linear regression was good enough. And Uma worked out or put together the mechanism based on various different literature findings to come up with this network and then analyze this network mathematically. We are almost out of time. And essentially write mass balances and rate equations and so on and so forth, and analyze this to get traces, rather variations that are predicted by the model. They seem to be close enough to the experimental data for the purpose at hand. This is a model anyway, so this seems to be fine. This many values were done. And then she went on to look at this aspect. I mean, this was my idea. The rhythms are not considered in cancer treatment management. Okay, so if you add an RS causing drug, ROS causing drug at the wrong time, maybe it's not that effective. That was the thinking, but we went about properly studying that. And since you do that, the undesirable side effects are also high. My thinking was, if you added at the right time, at least the side effects would be much smaller. And we showed for the first time that the phase of the reactive species rhythm at which the drug is added is significantly important in determining the cytotoxicity of anti-cancer drugs such as Minadion and curcumin and a few others in 2 different cancer cell lines as given in the paper. Okay. From that time onwards, for the past many years, we have been trying to get this to the hospital. It came to the stage of setting up meetings with surgeons, cancer surgeons and so on, but they are very busy people. That guy called, we set up 3 meetings and that guy called at the last minute in cancer doctors. So I said, okay, whenever it happens, and let us save it. This happened because a friend wanted us to talk and so on and so forth, looked at possibilities happening. This is the person in Apollo, but they are very busy. So they just have to try it out. There are no approvals needed here, right? They are all approved drugs. So they just have to try it out at different times. So cytotoxicity depends on the time of drug administration. This is a good social contribution with huge societal relevance. It was picked up by the newspapers and so on and so forth at that time. 27% higher and so on. So those were the 3 papers. Any quick questions? We're out of time. We'll have to stop. A minute beyond. Okay. So please come for the Thursday's class. We'll play a game, and then Friday, yeah, mostly, yeah, we are done with this. So Friday you can take off, and then Monday come back again, read that, and essentially take off in the sense time given to you for reading. And that's the whole idea there. I need to, practically give you time also. So that is what that is. And then Monday we look at the incentive as well as the basis, the stories. Okay. Any other questions? Okay. We'll meet on Thursday then.